Hello everyone, I am Anik Saha, presenting our work STELAR-X, scaling coalition-based species inference to 100,000 species and beyond. I am thankful to my co-author Dr. Shamshuzzoha Bayzid, and we are from the Department of Computer Science and Engineering, Kuwait. First, let's look at the problem we are trying to address. Let's say we are given page increase G1, G2 up to Gk, and our goal is to infer a species tree SP over the n species. However, the key increase themselves might be discordant from each other, and therefore we aim to infer a species tree that maintains a maximum consistency with the input key increase. So these are called summary methods, and importantly, summary methods are statistically consistent when done in terms of triplet or quartet consistency. Although summary methods are quite a common and powerful paradigm for addressing this problem, scalability still remains a major challenge. For instance, the current state of the art is STELAR-MP, which can analyze datasets of only up to 10,000 taxa and 1,000 key increase. And to analyze such a dataset, it requires about 16 hours of runtime and 124 gigabytes of CPU RAM. And based on projections mentioned in the paper, analyzing even only up to 20,000 taxa would require prohibitive amount of resources, potentially several days of runtime and hundreds of gigabytes of RAM. So this presents a serious challenge for modern phylogenomics because the datasets are largely growing in size increasingly. Now let's look at where STELAR-X comes into picture and how we push the boundary of scalability. For instance, in the same dataset of 10,000 taxa and 1,000 key increase, STELAR-X completes it within only 77 seconds, which is 712 times faster. And notably, it takes only 16.4 GB of RAM, which is pretty common even in our laptops. And it is 7.5 times more memory efficient than STELAR-MP. Well, not only that, it can also push beyond the existing limits. For instance, it completes a dataset of 100,000 taxa in just 8.5 hours, and most astonishingly, another dataset of 100,000 genes in just 4 minutes of runtime. And apparently, since these resource requirements are not very practical, even beyond 100,000 taxa would definitely be practically feasible within modest resources. Now we present a high-level overview of STELAR-X. First, we start with the input key increase. From there, we extract the sub-tree bipartitions. And then we construct a frequency map. That means we first identify which are the distinct or how many distinct bipartitions are there. For instance, here among the 6 bipartitions shown here, let's say we have only 4 distinct bipartitions. And then we also count, for instance, for the middle 2 bipartitions, they each occur twice over all the input key increase. And then comes the computationally most demanding step of our algorithm, which is the pre-computation of weights. So we also have a set of candidate bipartitions. Candidates are those bipartitions from which we choose while constructing the output species tree. And once we construct a weight map, which is a weight for all the candidate bipartitions, we then run a dynamic programming algorithm to obtain our output species tree. At this point, we gradually explain all the components one by one. Well, first, what is our inference objective? The inference objective of STELAR-X is similar to STELAR. We aim to construct a species tree SP that maximizes triplet agreement with the input key increase. That means we want to maximize the number of triplets that are common between our inferred species tree and all the input key increase. So now let's look at the dynamic programming algorithm that was also in the precursor STELAR. So if we look at the right picture here, it is very apparent that if we want to construct a tree, we eventually need to, we will definitely start with a cluster of all the n species, and then we will gradually need to partition the cluster to all the way bottom. And until we reach the leaves where there will be only single taxons. For instance, consider a cluster A. there might be many ways to partition this cluster into two different parts, A prime and A minus A prime, and we will somehow need to choose only one way between them, among them. So, well, how do we do that? Here you can see weight term Wg A prime bar A minus A prime, which represents the weight or the value we obtain from this particular partition. And then the dp recurrence is shown here, where we actually want to maximize the score for the cluster A. And how do we do that? We actually do standard 3dp, where the score for A is constitutes of the score for A prime, the score for A minus A prime, and importantly the weight of this particular partition, A prime bar A minus A prime. So it's very apparent that the expensive part here is to compute, store, and query the weights of all the candidates. Now we come to the first point, where STELAR-X majorly differs from the precursor scalar. The one particular bottleneck of the traditional algorithms, scalar and astral, is their representation of the bipartitions. Traditionally, each cluster in a bipartition is represented by an n-bit vector, where if a species is present, its bit is turned on, and otherwise the bit is turned off. The problem is that even if the cluster is very small, for instance, even if the cluster has only a single taxon, even then it would be a big set of length n, which is order of n space. And since we have order of n k bipartitions, and if each of them takes order of n bits, we would eventually need order of n square k memory. And this is one particular reason why the traditional methods were so much prohibitively memory were consuming so much prohibitive amount of memory. Now we look at how STELAR-X aims to solve this problem. Well, we actually introduce a novel representation, that is an integer tuple-based representation of a bipartition. Please note that for any tree, a bipartition or a subtree bipartition always corresponds to one particular internal node. And if we list all the taxa of the tree from a left to right order, we obtain the pre-ordered array. And importantly, each internal node induces two adjacent sub-arrays of that traversal array. For instance, we can also see in the right pictures that the bipartitions B bar B and the bipartition C bar A, both of them represent two adjacent sub-arrays of this array. And now, since we can store the pre-ordered arrays in only order of n space, and we can actually reuse these by only storing the indices of the sub-arrays to represent the bipartition. So how do we do that? For instance, if we look at the picture at the bottom, the subtree bipartition is C bar A, and C can be represented as a sub-array of 4, 4, assuming a 1 base indexing, and the A can be represented as a sub-array of indices 5 to 6. We also store which particular gene tree this bipartition came from, therefore we also store the gene tree index 3. And what is the result? The result is that this brings down the memory requirement from order of n square k to only order of n k, which is asymptotically optimal. That means as long as we can support the input gene trees, we can also afford running the entire algorithm due to this novel representation. Now, if we recall from our overview of STELAR-X, the next step was to recognize equivalent bipartitions and to detect which are the distinct ones. So what does equivalence mean? For instance, here A bar C and C bar A, they are basically the same bipartition because order does not matter here. Now, how do we test whether two of them are equivalent? Well, then we will need to test whether two sets of taxa are essentially the same after shuffling. But we could do that with a naive set comparison, but that would require scanning all the taxa one by one, and it would cost order of n time per comparison. But STELAR-X instead maps each bipartition to a compact integer hash signature, and then we simply compare those hash values. But importantly, our hash function needs to fulfill two important properties. The first one is to be permutation invariant. Naturally, since our two equivalent bipartitions, they do not care for the order their taxa are written in. So therefore, our hash function also needs to be permutation invariant. For instance, the hash for E, A and the hash for A, E, these must be the same, because otherwise they will be treated as different bipartitions. One more subtle property is to be associative. Well, why do we need an associative function? Because recall that we represent bipartitions as subarrays or intervals of the array. That means we will frequently require to calculate hash values of subarrays or intervals. If our hash function is an associative function, for instance, summation or in-store function, then it enables us to compute prefix hashes. And with prefix hashes, we can actually compute interval signatures in only order of 1. So it is basically the same idea of computing the interval sum from prefix sums in order of 1 by simply subtraction. And in STELAR-X, we use two different hash functions, summation and in-store. Both of them fulfill the two properties mentioned before. They are both permutation invariant and they are associative. So one question is, why do we use two different hash functions? Well, whenever we talk about hashing, a natural problem that occurs is collisions. So the use of double hashing makes accidental collisions negligible in practice. In the right picture, we show an example of hashing where we see that the partitions EA bar C and C bar AE are mapped to the same double hash, and therefore, when mapping the frequency, we can directly identify that yes, the bipartition EA bar C occurs twice across all the gene trees. Now our next important step is to calculate weights of bipartitions. Well, what does a weight of a bipartition X mean? It means how many triplets are compatible with X that are in the gene trees. So the number of gene tree triplets that are compatible with X. Well, how do we compute the weight of a bipartition? Recall that we had all the frequencies of all the gene tree bipartitions Y, and therefore we can simply sum over all the unit gene tree bipartitions and multiply that frequency with the particular triplet contribution between what that bipartition X and the gene tree bipartition Y. Now, how do we compute this MXY term? Well, it's based on simple combinatorics, where we derive this formula based on the overlapping parts of the two sides of the bipartitions, and the formula is presented here. One very important and interesting observation is that the DP repeatedly needs the weights for tangent bipartitions, and more importantly, the weights are not redundant. That means all the weights are needed by the DP state space anyway. A natural conclusion is that we can simply pre-compute all the weights. And therefore, in contrast to STELAR, in STELAR-X, we actually divide the computation into two stages. Before the DP, we pre-compute all the weights of bipartitions, and during the DP, we simply do a fast scaling lookup to retrieve all the weights. The next important thing we do at STELAR-X is parallelization. In CPU, we simply assign the input gene trees in batches to CPU threads, and for parsing or subtree bipartitions, since they are independent across all the gene trees, they actually run in parallel across the batches of input gene trees. Most importantly, our most computationally intensive part, weight computation, is actually handed over to GPU. Recall that we had to determine the MXY contributions between two bipartitions. And since all these pairs of XY are essentially again independent, each GPU thread independently handles a distinct bipartition pair XY. Then the results are reduced to obtain the final weight map. The resulting time complexity and memory complexity are shown here. Very importantly, the running time, the dominating part of the running time complexity is the n square k square by tg, where tg is the number of GPU threads. Naturally, this part comes from the pre-computation of weights. And notably, the CPU memory and GPU memory complexity both actually simplify to order of nk, which is asymptotically optimal since our input size itself is order of nk. So here are again some scalability results from our study, where we see that in the 10,000 extra dataset and in the 10,000 gene tree dataset, in both of these cases, STELAR-X is much more time and memory efficient than STLMP, and more importantly, STELAR-X can even push beyond the current limits and can analyze datasets of up to 100,000 extra and 100,000 gene trees. Importantly, although STELAR-X pushes the boundary of scalability, it also remains competitive in terms of accuracy. It is comparable to leading methods STLMP, WTSMP, and STER. As shown in the first three panels, and in the last panel of the figure, we also see that it matches or exceeds STLMP, and it also exceeds STER in several large simulated datasets. We also, side by side with the simulated dataset, we also analyze biological datasets, and here we show the results from avian dataset, where the similar plates are shown in the same color, and we can easily interpret that the STELAR-X is recovered evolutionary history that is broadly consistent with both the published reference and the result from STLMP. And here we again show that our inference was very efficient, and in avian dataset, our inference was 30 times faster than STLMP, and in the extended avian dataset, which consisted of 63,000 genes, we completed that 7.9 times faster than STLMP. To conclude, STELAR-X is the first statistically consistent method to analyze at the 100,000 taxon scale, which is a major leap forward. And notably, it does it within practical time and memory requirements. So this matters a lot in modern phylogenomics because researchers are increasingly being interested in large tree-of-life initiatives, for instance in resolving the angiosperm tree of life. And if we integrate STELAR-X into divide and conquer frameworks, we believe it can push inference toward millions of taxa, and that will eventually be able to support such large initiatives. So thank you.